Welcome, everyone. I'm Agnese Winfield, your moderator for today. I'm NanoString Marketing Director in EMEA, and I'm really happy and excited to introduce the webinar today. We are focusing on neuroscience today. Today is the third day. It's the third day of the spatial answer trilogy. We focused on oncology on Tuesday. Yesterday, we focused on immunology, and today is the turn of neuroscience. Please note that this is a live webinar, so you can really post your questions. There is a Q&A box there on your screen. Please use it, and we'll be very happy to answer your questions. Our guest speakers will be taking them on live at the end of the presentation. So the session will run as follows today. First of all, we have about 10 minutes of a technology introduction. This will be presented by my colleague Tony Zucker from NanoString, a specialist of geomics, who will introduce the spatial genomics revolution with the Geomix digital spatial profiling platform, followed by our very nice colleague and friend, Pavel, from the Illumina company, who will talk about and complete the workflow of spatial genomics. After these two presenters, I will introduce you to our guest speakers. For now, I just tell you that one of them is Professor Philipp Khaitovich, professor from the Center of Neurobiology and Brain Restoration from the Skoltech Institute of Science and Technology in Moscow. And the other presenter is Joan Liu, Senior Research Fellow from UCL Queen Square Institute of Neurology in London. So now over to you, Tony and Pavel. Thank you.
Great, thank you very much, Agnese, and thank you everyone for joining us. We're really happy to be here, and we're looking forward to showing you some, we have some great guest speakers here with some great data. I'm going to start off a little bit about the Geomix platform, our spatial technology. And the Geomix and NanoString as a whole, we really have a way to make the biology simple but give you incredible data. We've gotten over 4,000 publications with 1,700 different signatures, and we're really proud of that. But finding the right biomarker can be really difficult. How do you do it? And classically, especially with the nCounter, NanoString's initial technology, it's really powerful with this example that was done in collaboration with Merck, where it was bulk expression, found an 18-gene signature, really important for a companion diagnostics, and they were thrilled about that. But if you look in the bottom right here, there were still some genes, some patients that had an upregulated signature of these 18 genes, but still didn't have improved progression-free survival. And why is that? What could help tease that out? Well, we think it's spatial gene expression, where you can draw an ROI around highly hot but excluded tumor, highly infiltrated tumor, or maybe that bulk gene expression would struggle, but you can really see the differentiation with the geomics and the spatial capabilities. What the geomics can do at a high level is fresh frozen, FFPE, RNA and protein, high-plex capabilities of 18,000 gene whole transcriptome with human or mouse, 100-plex protein, and an ROI selection that's quite tunable. There's no trade-offs with the geomics, but how does it work? Well, it works by having photocleavable linkers, and we can attach that to antibodies, we can attach that to RNA probes. When UV light hits this, these probes are released into solution, and we capture them and then count them. With RNA, we are doing basically for the whole transcriptome 18,000-ish experiments all at once, which means we're really robust in FFPE. Of course, we can do fresh frozen. That's the easy stuff, but we're not doing poly-A capture. We're doing an unbiased, targeted approach, and work really well in these difficult matrices. The whole process goes where we have those photocleavable linkers to either attach to an antibody or attach to an ISH probe. Then we mix them with fluorescent antibodies, which I call fluorescently labeled morphology markers, because you mix the profiling reagents with these morphology markers, and then number 2, you select your ROIs based on the morphology, illuminate with UV light. You then in number 3 will cleave those probes, and then you go to the next ROI with a new fresh buffer, and then you release those probes, and everything is collected into a 96-well plate, where that plate goes downstream to be counted, and then you bring that back to be analyzed with some really interesting software tools on the Geomix. As a whole, what the Geomix is, its beauty is its simplicity. You're getting this whole transcriptome or 100-plex information, but it's all simplified by defining your ROIs early. You can decide exactly where you want your data to come from. You could be comparing astrocytes around a particular plaque or a particular cell type, but because you defined it early, then when you start looking at your whole transcriptome information, the data is easier. In this example here, you're seeing a list of genes, and those lists come back to us with counts, and then each of these counts is then traced back to a particular ROI. And our software can work through this, and or if you have the biostats experience, you can work with that yourselves. And I really do think that simplicity of being able to define things early, also our reproducibility, is what's gotten us into so many journals in general in just the year and a half we've been working, but also high impact publications, Nature Medicine, Nature. And the last slide is just kind of a reminder. The strengths of this platform is fresh frozen and FFPE. It's protein, 100-plex protein, 1800-plex whole transcriptome human, 22,000 whole transcriptome mouse, tunable ROI selection, whether it's just an ROI, a circle, or if it's a single cell population that you want to illuminate, there's that kind of throughput. And definitely we can do 4 to 10 slides in a day. And then now I'll pass it over to you, Paul.
Thank you very much for this very nice introduction to the Geomix platform. And my name is, as I mentioned, is Pavel Sayak. I'm part of Illumina's marketing team, and it's my great pleasure to, within the next couple of minutes, briefly introduce who we are and what we offer. And as Tony mentioned, the readout for the high-plex Geomix assays is done on Illumina sequencing systems. And this is a truly exciting field. It was selected as the Nature Method of the Year just a couple of months ago, and it really shows how exciting this technology is. So biological systems, organs, they of course have a precise organization because it's important for their function. And being able to look at expression within this tissue context is truly valuable because it gives an additional layer of information, and it can really tell you more about the underlying biology. And I wanted to start with a brief introduction of Illumina. So for those of you who haven't heard about us, we are a global company, and we develop, manufacture, and market innovative next-generation sequencing and microarray technologies. And our technology enables you to make foundational discoveries, but also to transform medicine and ultimately to impact lives. And there are more than 200,000 publications where our technology has been used, so it clearly shows that this is a robust technology, giving you data you can trust. And you can either purchase a system and implement it in your own lab, in-house, or you can also turn to one of the many service providers found throughout the world and get access to our technology there. And our sequencing systems, they are a very flexible tool. And depending on how you prepare your samples, or how you do the library preparation, you can use the sequencer to study several different types of biological molecules. So you can study different facets of DNA, you can study different populations of RNA, you can do studies at the single cell level. And as Tony very nicely illustrated, you can also do spatial profiling, look deep into tissues with retained spatial information. And our systems fits very nicely with the output needs of the nanostring geomic DSP assays. So for example, the larger Geomix assays, the cancer transcriptome atlas, allowing you to look at 1,800 genes, and the whole transcriptome atlas, allowing you to look at all 18,000 genes, they are an excellent fit to our NextSeq and NovaSeq sequencing platforms. And of course, data analysis is also an important step, and we want to make it as streamlined as possible. So now, on Base-Based Sequence Hub, which is our cloud-based environment for sequencing data storage and analysis, there is now a Geomix Pipeline. So this application, this app, will basically take the sequencing reads, and it will convert these, analyze these into a digital count conversion file. And you can then take this file and load it onto the Geomix DSP instrument to do spatially resolved analysis and different types of visualizations. And of course, if you have any questions about our sequencing solutions, our technology, please don't hesitate to get in touch. So with this, thank you very much for joining us today, and I will hand the word back to Agnese.
Thank you very much, Pavel. Thank you, Tony. And we just proceed on, and I'd like to introduce our first guest speaker, Professor Philipp Khaitovich, who I invite on the camera now. Let me tell you a little bit about the biography of Philipp. So Philipp completed the undergraduate studies in molecular biology at Moscow State University, followed by a PhD in biochemistry at the University of Illinois at Chicago. Philipp worked as a postdoctoral researcher at the Max Planck Institute in the Department of Evolutionary Genetics, till he took a faculty position at the Institute for Compu-tational Biology, jointly established by the Chinese Academy of Science and the Max Planck Society in Shanghai, in China. Philipp was then promoted to the Institute Director position, and from 2012, Philipp is a fellow member of Max Planck Society of Germany, as well as adjunct professor at ShanghaiTech University in Shanghai, China, and from 2014 is also full professor at the very famous Skoltech Institute in Moscow. And the presentation today is the transcriptomic map of schizophrenia. Thank you, Philipp. Take it away.
Thank you very much, Agnese. Today I will talk about the transcriptome map of schizophrenia. Schizophrenia is a disorder which needs no introduction. Unfortunately, it affects about 1% of human population. But still, despite decades of research, we don't know the mechanism of disorder to detail, and because of this, we cannot really design good treatment or good prevention therapies. And part of the reason we cannot really easily identify the causes of schizophrenia because it affects the brain, and the brain is incredibly complicated tissue which contains about 100 billion of neurons, and each neuron connected to the other neurons by 1,000 to 10,000 connections. So when some people compare our brain to computer, it's totally an underestimate. We should compare our brain to a huge internet with maybe 100 billion users. Of course, it makes it very difficult to identify the reasons for any disorder, and this includes molecular causes, which could be reflected by gene expression. So in our work, before we turned to the Geomix analysis, we did some background work. We used conventional RNA sequencing to build some kind of equivalent of transcriptome map of the schizophrenia controlled brains. We took number of brains from schizophrenia individuals and control individuals. And here you can see the actual slices we used. Some of you heard about gray matter and white matter of the human brain, and you can see it really exists. This is a slice of frozen brain from collection which we used, and we talk about 35 different functionally and anatomically different brain regions. Here you can see the map of these regions. So there were regions representing our cortical areas, which shown here in blue, dark blue, green, and light green. But there were also cerebellar regions, subcortical regions shown here in red, white matter regions shown in purple, and some deep nuclei file you can see here in orange. And we did conventional RNA sequencing to get the data and then compare it, did statistical analysis, and I'll show you the main result of this analysis. It is here. I apologize for the quality of slides because it's work in progress. Perhaps it's a little bit too small to read, but immediately what you can see, with 35 brain regions listed here, and here the bar shows just number of differently expressed genes, also with direction, but direction here is not so important. More important the numbers. You can immediately see what the numbers are very different, and among brain regions. And actually there are some favorite schizophrenia brain regions most neuroscientists use. These are Brodmann area 9 and Brodmann area 24, with parts of the frontal cortex here and the cingulate cortex, which is in between two hemispheres. But actually, if we look at them, we will see very few differences. So with letters here, if they're not too small for you to read, with BA9, you can see it's here, and other BA areas, this is just Brodmann areas of the cortex. The neuroscientist who named this areas lived about 100 years ago, and he distinguished these areas based on their histology composition of different layers of the brain, because our cortex is just a layer cake. And this separation still makes sense because with different areas, they play different roles in our brain. And you can immediately see what among all Brodmann areas, there's just one with green one, which stands out. This is Brodmann area 22, the one is here in the temporal cortex, and all this frontal cortex area and parietal cortex area being light blue or dark blue. They don't really show as many differences as one would expect, because till now, if you look at the bulk of transcriptome studies of schizophrenia, maybe 90% of the papers would examine either Brodmann area 9 or some other part of the frontal cortex. We also find lots of differences in amygdala, and this is rather expected. So neuroscientists already knew what amygdala is involved in schizophrenia. But we also find a lot of differences, and also in both cerebellar regions. And this is really not something very well studied. So already here we see quite a lot of new things. But of course, for us, most interesting to investigate things which happened in the cortex. And so we looked at the two cortical areas. Brodmann area 9, which is the one which has very few differences in our analysis. And this actually agrees well with previous data, because even though many people looked at it, frequently different labs get different results. And this is not so surprising, because if you have very few small differences, it's hard to reproduce. And Brodmann area 22 in the temporal cortex, it's not really investigated in schizophrenia at all. So we, with the help of NanoString, which we are very grateful for, we were able to take a look at these two areas and not only investigate the bulk expression, of course, because it's spatial, but we were able to see expression in different layers of the cortex. Remember, I told you it's like a layer cake. And so there's 6 different layers. Neuroscientists usually distinguish from the bottom to top: 6, 5, 4, 3, 2, and 1. Overall, there's quite a lot of genes could be detected, but we selected very strict cutoff, which from around 18,500 genes overall detected. We looked at about 1,200, because we wanted them to be highly expressed, which is still a very big number of genes, considering what this is spatial analysis. And first we compared the differences which we see, not the expression of genes, but it's important we compare the differences which we see between schizophrenia and controls in two brain regions in Brodmann area 9. You can see it's here, prefrontal cortex, this is a little brain, and this is frontal part of it, and the Brodmann area 22, which is here, part of the temporal lobe. And why it's important? Because if we look at the papers, even using bulk RNA sequencing, just the conventional RNA-seq, very frequently there is no correlation between the differences reported by one study and another study. And part of it because in some regions there are a few differences, I said, like Brodmann area 9. And indeed here we see quite weak correlation, but still significant. So, which is very good. It means what, despite the fact that there's very few significant differences, we still can reproduce them using nanostring technology. But Brodmann area 22, which shows more differences, of course, it looks much better. So the correlation between the bulk RNA data and the bulk nanostring, this is now pulling data from different layers into this kind of pseudo-bulk expression. The correlation looks quite obvious, and of course it's highly significant. But more important for us, do we see different number of differences, so to say, in each layer? For instance, because we know different layers of the cortex, they have very different function in brain, very different histological architecture. And indeed we see that. So if we look now more closely, this is now number of differentially expressed genes in different layers from 1 to 6 in the Brodmann area 22, shown here in green, or Brodmann area 9, shown here in light blue. And you can immediately see what actually, even though in Brodmann area 22, we obviously see more differences, and this is now just nanostring data, this is Geomix whole transcriptome sequencing, and this is a significant differences, this is not some kind of detected genes. So we see the significant differences, statistically significant differences, and we see, first of all, that in agreement with our bulk transcriptome analysis, Brodmann area 22 shows more of his differences, but we also see what most of his differences, at least the majority, are in upper layers, in layers 1, 2, and 3, especially 2 and 3. And in a way, in the Brodmann area 9, we see similar pictures. There's also some differences in area 4, but at least layer 6 and layer 5, to some extent, they show much fewer differences. Then the interesting analysis. Again, I apologize for such small figures. I hope you can read them. But this is cell-time composition analysis because we know what brain consists of different types of neurons, excitatory, inhibitory, and astrocytes. And here we were able to look at the marker genes, which are characteristic of astrocytes, of excitatory neurons and inhibitory neurons. And here the stars indicate significant differences in schizophrenia. And one thing which we clearly can see with astrocytes, so they're called astrocytes because they look like stars and they support the neurons, they really very clearly and obviously decrease in the middle layers of the cortex. But the neurons, both excitatory and inhibitory, they increase. So the differences which we see there, especially in the layers 2 and 3, it's a combination of signals which we get from astrocytes and from different types of neurons. In addition to transcriptome data, we were very lucky what nanostring helped us to generate the proton data. And so we also looked at these two areas at the proton level. And here it's a different order, so don't get confused. This is the region 22 and region 9, sorry. This is built in the opposite order, but nonetheless, the result is the same. We see more differences in region 22 compared to region 9. You can see the layer distribution is a little bit different. So there in for RNA, it was area layer 2 and layer 3 mostly, and here's layer 1. But this is also very important and interesting because in the cortex, frequently cell body is sitting in one layer, but the protrusions of the cells, for instance, axons or dendrites of neurons, which contains a lot of proteins, they actually can extend and do extend to other layers. So here the discrepancy between the RNA and protein data is an additional source of information. And the last thing I wanted to show some rather striking examples of differences which we were able to see based on the protein data for some very well known neuronal markers such as vimentin. So here you can see the control in schizophrenia, and here you can see the layers 22 and the 9, and the differences in the layer 22 and much bigger than in layer 9, but actually layer 9 also has differences. So people who were studying it before and finding differences there, they were not wrong. Just differences in the layer 22, the region 22 are much larger. We can see decrease in schizophrenia compared to control. Vimentin, which is a marker of specific neuronal marker of particular cell types. And also very interesting discovery for the myelin basic protein, because there are some indications of hypermyelinization of area 22 based on the functional MRI data. And we can see actually very, especially in superficial layers, and we can see very strong increase in schizophrenia of this myelin basic protein, which involved in this myelination of axons. And this matches very well published data, and we're now also going to do additional experiments just to explicitly look at this white matter component of the cortex using multi-imaging, which enables us to visualize lipids which are there in the myelin, which is would be then another technology, another spatial technology which will complement this one. And hopefully this is a result which will be able to support by additional data. So this is all what I wanted to show you today. We will definitely have more results out of this wonderful data set, but this would require additional experiments which we are now doing. And I would like to thank NanoString for enabling this work, and of course to our colleagues from the psychiatry centers, which provided us with samples which we used in this research and their expertise. And thank you all for your attention.
Thank you very much, Philipp. Great presentation. We're going to take the questions after the next presentation, so feel free, everyone, to post your question in the Q&A box that you see on the side of your screen. Philipp, we are going to take now a poll. You can rest for a minute, don't go away completely, and we'll see you for the questions later. So we're moving on a quick audience interaction. So if you would like to participate and take the next poll, it would be wonderful. We would like to hear from you when you conduct spatial research, if you're not doing it now, in future. What analyte will you focus on, or are you focusing on? Is it RNA, is it proteins, or is it both? And we've run this same poll in the previous days, and we found very different answers, actually, from the oncologists versus the immunologists that attended the previous two webinars. So it would be nice to see the results now live so that we can share with you those as well. Okay. Another different question. So we see that RNA and proteins have been selected by all of you, which is very, very interesting. Excellent, excellent. Thank you very much. So I would like now to introduce our second speaker. It's my pleasure to invite on camera Joan Hugh, who is the Senior Lecturer in Cellular Pathology at the School of Life Sciences, University of Westminster, and an Honorary Senior Research Fellow at Division of Neuropathology, UCL Queen Square Institute of Neurology. Joan's research investigates the pathomechanism underlying structural pathologies, inflammation, and neurodegeneration in human epilepsy. And the title of the presentation is Single Cell Expression Studies in Human Focal Cortical Dysplasia. So Joan, thank you very much for being with us, and we are all looking forward to listen to your presentation. Thank you.
Hello, everyone. Thank you very much, Agnes, for the introduction. I'm very happy to be invited here to present my gene expression study on focal epilepsy using nanostrands digital spatial profiling technology. I just want to acknowledge that this study was funded by the Horn Family Charitable Foundation through the Epilepsy Society, and the tissue used today were obtained from the Epilepsy Society Brain and Tissue Bank. Epilepsy is a chronic neurological disorder where patients experience spontaneous and recurrent seizures. And a lot of them also experience cognitive and memory impairments, which really affects their quality of life. There's no cure for epilepsy patients at the moment. For most patients, epilepsy can be controlled through anti-seizure medication. However, approximately a third of these patients, especially those with focal epilepsy, are refractory to drug therapy. So one alternative treatment option for these patients is epilepsy seizure, whereby an area of the brain that initiate abnormal seizure activations are removed, and the seizure freedom is again in approximately 60 patients who undergo these surgeries. Our research group is very interested in studying these surgical resections from epilepsy patients so that we can explore the pathomechanism underlying epilepsy. But for the present study, we are very interested to study specifically the brain resections from patients with focal epilepsy and also focal cortical dysplasia pathology. Focal cortical dysplasia is one of the most common pathology findings in surgical specimens of patients with focal epilepsy. FCD, or focal cortical dysplasia, can be found anywhere in the brain, with about 51% of the cases observing FCDs located in the frontal lobe. This type of cortical malformation can be categorized into 3 subtypes or tiers based on clinical pathological criteria set out by the International League Against Epilepsy Task Force. In particular, FCD2B is the specific pathology that we're interested in for this study. That's because FCD2B is characterized by really abnormal architectural and cytoarchitectural differences in the cerebral cortex. And our main pathological findings in these specimens include cortical delamination, radio, neuronal disorientation, poor myelation in the white matter, and the presence of abnormal cell type known as dimorphic neurons and balloon cells. And these are the abnormal cell types that we want to explore further on using nanostrings digital spatial profiling technique. Here you would see image showing an H&E or hematoxylin stained brain tissue section from a patient with focal cortical dysplasia 2B. And you'll see in A that there are a mark pale discoloration at the bottom of the sulcus where the arrow is pointing to. And the FCD is located particularly in the bottom of the sulcus for this patient. And it's actually quite a common location where FCD can reside in most patients. And in this region, you would see that the clear demarcation between the grey and white matter is lost. B is showing a really higher magnifying view of the bottom of the sulcus where the FCD is located in this patient. And on the either side of the sulcus, you would see that the normal six-layer cortex is actually disorganized now. And there's a lot of pleomorphic cells scattered throughout the grey matter. In particular, you may actually see these larger cells with larger, strongly stained hematoxylin stained nuclei clustering kind of in the middle and lower cortical layers, and an example is noted around where the star sign is located on the image. And there's many actually abnormal cells, huge cells, that are found in the white matter underlying the the grey matter that's located at the bottom of the sulcus, and this is where the triangle signs are located, pointed to in the image here. Let's have a look at these two locations of interest in a high magnification. Okay, so in C, you would see that there's a high number of abnormal neuronal cells known as dimorphic neurons. And these cells have a very large cell bodies and nucleus up to about 50 micrometers in diameter. And this is comparable to normal neurons, which are usually up to about 25 micrometers in diameters. And these dimorphic neurons are mal-orientated. They abnormally aggregate a lot of intracellular missile substance, which are displaced towards the cell membrane. And there's a lot of cytoplasmic accumulation of neurofilaments as well. In D, here you would see that there's a large number of paler, large balloon cells. They have this glassy, elfinic looking cytoplasm, and the nucleus is normally disorientated to the periphery. And sometimes these cells are bionucleated or multinucleated as well. So both of these abnormal cell types are really best appreciated using immunohistochemistry. And dimorphic neurons can be identified using a marker for neurofilament, for example, SMI32. And Vimentin antibodies can then identify some of the balloon cells that we see in FCD brain's receptions. In our previous studies, we've found that dimorphic neurons and balloon cells also express PES6, showing mTOR activation. But it should be noted that SMI32 and Vimentin and PES6 don't exclusively label these abnormal cell types. There's actually no single marker that labels these particular cell types. So that's why we want to use the, digital spatial profiling to investigate these cells a bit more. And also, despite the many kind of SCD studies around this area, we really don't know how these abnormal cell types contribute to epidermogenesis. And so we are very interested to use DPS to learn more about the gene expression profile of these cells and how they differ to the morphologically normal appearing cells in the nearby non-diaplastic region of these tissue. So to start our journey, we contacted our local NeuroString rep, Xavier and Tony, last summer to start the project. It was very exciting, but here's a brief summary of the workflow. So first we prepare 5 micrometer thin formalin-fixed paraffin embedded brain tissue from 4 cases of FCD patients who have similar FCD2B pathology. And in these cases, we see a lot of dimorphic neurons, particularly in the grey matter, and balloon cells, particularly in the white matter and also in the subpair as well. We sent our unstained tissue and morphological markers, SMI32 and Vimentin, to the NanoString team in the US. So, for example, Allison and her team was able to perform digital spatial profiling using a panel called the Cancer Transcriptomic panel, and this panel consists of about 1,800 genes. The US team labeled our sections using immunofluorescence and the marker that we sent them. And on these immunofluorescence labeled tissue, we then outlined our area of interest. And then we used a special segmentation method to actually segregate these tissues so that they can be further analyzed using DSP. And here's a bit more detailed about our cases. So we've got our 4 cases here showing FCD2B pathology. These cases range from the age of 20 to 40 at surgery. We tried to use tissue from more recent cases, so we included 2 cases from 2019, 1 case from 2017, and 1 case from 2014. And the resection were predominantly from the frontal cortex, with 1 case in the parietal cortex. Here's a closer up look at our tissue from our 4 cases. So So the dysplastic or SCD region is highlighted in yellow here, and these areas contain dimorphic neurons and balloon cells. And I've also noted where the non-SCD regions are located nearby to these regions. But the non-SCD regions contain morphologically normal neurons and glial cells, so they don't have the appearance of these abnormal cell types that we see in the FCD region. So we're interested in comparing really three groups or elements. First, we're interested in looking at the gene expression profile of the FCD region compared to the non-FCD region. Next, we're interested in comparing the gene expression profile of abnormal cell type dimorphic neurons compared with morphologically normal appearing neurons in the non-FCD region. And then thirdly, we're interested in comparing gene expression profile of balloon cells in FCD regions compared to morphologically normal appearing glial cells in the non-FCD region. To isolate these particular cell type and also region, we will need to perform specialized segmentation logarithm so that we can select these samples for DSP. And these are actually shown in image A and D. So for comparing FCD and non-FCD region, we just draw a circle in the area that we want to perform geometric segmentation on. And to compare specific abnormal, dimorphic, and balloon cell types are shown in B and E, and also normal appearing neurons and glial cells in non-FCD region are shown in C and F respectively. We draw three squares region in those places and to get sufficient tissue for our abnormal and normal appearing cells for digital spatial profiling analysis. And as detailed in the workflow, the actual selection of the areas of interest were conducted on tissue that were labeled using immunofluorescence marker against SMI32, vimentin, GFAP, and a nuclear stain as shown in A. And B is a figure showing FCD region, which is encircled for the geometric region, really for the geometric segmentation. And then I've also included a square here to show that that's the area that were used for rare cell segmentation. And C is further showing how SMI32 dimorphic neurons are segmented from other cell type and tissue areas in that region, so that DSP is only performed on SMI32 cells and processes from the FCD region, as shown in this image here. NeuroScan has a DPX data analysis platform, which we can then visualize our data. The platform is really user-friendly, and you're able to actually select which sample you want to visualize or analyze. And the data we received has been through sequencing and biological probe quality control and then normalized by the NanoString team. So all the graphs presented here today are actually generated from the digital spatial profiling data analysis platform. First, here showing our principal component analysis plot. This is really interesting finding because our samples, as you can see, segregated quite clearly into 4 distinct clusters. So we've got the dimorphic neurons clusters, the balloon clusters at opposite end from each other, and then we've got distinct clusters of morphological neurons and glial cells. When we plot our data from the tissue that's been only analyzed using the rare cell segmentation program, we can still see these 4 clusters very distinctively in the PCA plot. But when we look at the geometric data, so this is just looking at region and not looking at cell-specific information. The separation between the four groups were not so distinct. So for the rest of the presentation here, I'll present data only from the cell segmentation program. We further did spot plot to check to see if markers we expect to be highly expressed in neurons were really indeed highly expressed. And here we can see that the neuronal population, dimorphic neurons and the morphological neurons were highly expressing neurofilament light chain, which encode for neurofilament, a form of it. And the expression in these cell types were higher compared to the balloon cells or the glial population. And similarly, when we look at CAM kinase 2B, we see also a higher expression of this gene in dimorphic and neuronal cells compared to balloon cells and glial cells. Looking at more kind of glial marker, S100B genes were highly expressed in kind of the glial population of balloon cells and glia. Then the dimorphic neurons and neurons. LAMP1, which is a lysosome-associated membrane protein, which were found to be highly expressed in balloon cells in previous studies. In this study here, we do see a high expression of this gene in balloon cells. And it was surprising to note that LAM1 expression was also higher in the dimorphic neuron population as well. We then perform heat maps, and we can see very distinct clusters, sets of genes that were highly expressed as represented in red and yellow here in the heat map. And these genes were mainly from the dimorphic neuron population. And it was interesting to see that the same set of genes were actually downregulated in the balloon cell population. And vice versa, when we look at the genes that were upregulated in the balloon cells, they were downregulated in the dimorphic neurons. We further did some more plots using volcano plots, and it shows kind of similar findings as seen in those heat maps. On the left, the plot shows kind of over 30 genes that were significantly expressed over twofold change in the dimorphic neuron population compared to the normal appearing cell or neuronal cells. And the right plot, you will see that there's over about 50 genes that were highly regulated or highly expressed in the balloon cells population compared to the glial population. And further pathway analysis showed us that these genes that were upregulated in balloon cells and also dimorphic neurons seems to involve kind of cell signaling pathways and also immune response pathways. And we're exploring these pathways in more detail at the moment. So in conclusion, this DPS study is very exciting, showed us very interesting and also surprising data as well. But it does actually tell us that the gene expression profile of transcriptomic profile of dimorphic neurons and balloon cells in STDs are likely to be distinct and different from each other, and also from the morphologically appearing neurons and glial cells in non-STD region. These changes in gene expression seem to suggest that there's some signaling pathways and also immune response pathways are involved. And our next step is to validate the digital spatial profiling findings through RNA-scope or RNA-ish immunohistochemistry. We have already begun testing markers that we can get and perform using immunohistochemistry, and we do see a high expression of these some of these genes that were upregulated in DPS and immuno studies as well. So it's very, very exciting. And we do plan to do more DSP, probably using the WTA panel, and it'll be very exciting to see what kind of results we get there. Okay, thank you everyone. Thank you very much for your attention.
Thank you very much, John. That was another great presentation. So I think we could take another moment to run another poll quickly, and then we'll come on all on camera to get the question and answers. There is a little bit more time also to post your question if you have any. Please use the question box in the screen. And on the screen now you have the poll. What type of experiment do you consider essential to enable you to answer your scientific question? So is it the bulk analysis, is it the single cell, or the spatial profiling? If you would like to take the poll now, it's open, and we can see the results in a second too. Okay, not many participated on this one, I don't think. So maybe we can take the poll away and then go to the question and answers for our presenters. If you would like to come back on camera, Tony, Philip, and John, I have some questions for you that come from the audience. So let's start. Let's start with you, Philip. So did you find a good agreement between the differences that you see in the schizophrenia at the protein and RNA level? So what was the question there?
Yes, Agnes. So indeed, we overall found good agreement, but it's actually more than that, because in the brain, the RNA and the proteins are frequently in the different places because cells are so extended over the layers. So we actually saw several good examples. So for instance, the myelin protein, we saw what most of RNA differences are in the deep layers, but most of the protein differences are actually in superficial layers. And now we're following up on that, and we see what this is actually the actual difference between RNA and protein expression in the schizophrenia brain compared to controls.
Okay. Actually, that leads me to the other question that we receive around your strategy for the segmentation when you use Geomix. For the reason that you just explained, the cells are so extended. What was your strategy in the segmentation?
Well, our strategy was very simple because we actually simplified this for ourselves in terms of choosing neocortex of the brain, and there's very well known anatomical segmentation in 2 different layers based on the cell histology, and we simply follow this classical neurobiological segmentation, which already exists for more than 100 years. And based on this segmentation, we really do get very different expression profile and very big differences among layers in terms of schizophrenia effect.
Yeah. Did you, last question for you before we move to Joanne, what type of tissue did you use? Did you use fresh frozen or was it fixed?
It was fresh frozen tissue.
Okay, perfect. Same question to you, Joanne. I think you mentioned, what type of tissue did you use?
We used human tissue, and they are formalin fixed paraffin embedded tissue, so FFPE tissue. And this was quite important for us because we are interested in using fixed tissue for to trial these kind of molecular analysis because FDD is actually quite difficult to localize in fresh or frozen resection. And a lot of the FDD is actually confirmed through immunohistochemistry on fixed tissue using a panel of different markers. So we definitely was very interested in using FFPE and see how DSP actually perform using this type of tissue.
Yes, and did it go well, I guess?
Obviously. Yes, and we're very happy. Yeah, definitely very happy with our results. And it's very interesting as well.
And again, on the sample, so talking about the strategy for segmentation, would you recommend using the rare cell segmentation over the geometric segmentation? What is your view?
Yeah, definitely. I think the rare cell segmentation really strongly popped out to us. When we saw that, it was, a bulb light up, and we were really excited to try this. But I think depending on what your researching question actually is, will determine what kind of segmentation you want to use. With our STD specimens, we have very good, distinct cell type that was abnormal in STD, and we want to really research on these cell types specifically. So using the rare cell segmentation is really the best way to segregate these cell population from other tissues and other cell types. In previous previous studies, we have used laser capsule microdissection and proteomics using frozen tissue, but that actually took a really long time. And actually using the DPS and also the rear cell segmentation, that makes the whole study really more efficient. And I think, because we've been through the whole process ourselves with Alison and the NanoStrand team over in the US, we are really more certain of our results and what cell type are being picked up and analyzed in DSP.
Okay. We have a technical question, Tony, around the slides that are to be used with Geomix. Are they special slides? And what type of slice also from FFP? What thickness?
Yeah, sure. So they are not special. It is a classic Superfrost Plus slide, which can be found really almost anywhere in most pathology labs. If you're using a like a bond system or have kind of automated, then we'd recommend a like a bond slide. And then the thickness, we recommend between 4 and 7 microns for tissue sections.
Perfect. Joan, another question for you. What is the clinical significance of your findings from these DSP studies?
Yeah. So STD2B, like I said before, is really a common malformation pathology that's observed in surgical resections of pediatric and also adult patients with focal epilepsy. And the previous studies have shown that dimorphic neurons and balloon cells are suspected to play an intrinsic role in mediating epileptic seizures. And our studies have shown that they're activated through the mTOR pathway. So getting to know the genetic profile or the transcriptomic profile of these cell type would give us important insight into a signal transduction underlying the cellular morphological differences that we see in these cell types and how it could contribute to brain malformation, and hopefully subsequently helping with development of kind of neurotherapeutic interventions. And secondly, like I said before, there's no really exclusive marker that labels dimorphic neurons and balloon cells. And hopefully by looking at those results that we've got and analyzing through very carefully, we can have a kind of transcriptomic profile of these cell types that can be used and be very helpful for classification.
Okay, last question for Philipp. Same question, actually, the clinical significance of your findings from this experiment. Can you see a path to the clinic?
Well, I believe we hope to have one. Of course, it's a very basic science study looking at the mechanism of schizophrenia, but it does help to look deeper. So, for instance, we already saw that it's much more effect on many brain regions which were not involved before in disease, implicated before in disease. And now, with the help of NanoString, we also see which layers have these effects and whether they are replicated across brain regions. So it does help us to build a more systematic view of the mechanism of disease. And this would lead us to the potential genes, which might be key regulators. And I didn't show it at today's talk, but we indeed identified a number of key regulators which might be guiding this expression difference which we detect. And they were picked up by the genome-wide association studies previously. So it is potentially an important study, and we hope it would shed light on the schizophrenia, which would allow us to move much further beyond what we can do now.
Wonderful, wonderful. Excellent. So I want to thank everyone here, the speakers, all of you. Pavel, we didn't have a question for you. I guess Illumina is known to everyone. So thank you very much to the speaker. Thank you to the audience who has been following us. And we can now close. We have a short video to say goodbye to you. Thank you. Bye-bye, everyone.
Bye-bye.
After completing slide preparation, which is similar to IHC or ISH, we will put our slides into the tray, and then we'll put the tray into the instrument and load buffer. Next, we will be ready to enter our slide records and scan parameters. A fluorescent image with up to 4 colors is generated to enable selection of region of interest or ROI on this tissue. You as the user can choose these ROIs from anywhere on your laptop. Geometric profiling allows us to choose circles, squares, or even polygons. This would be a great tool when you have pretty homogeneous regions like immune cells and the tumor segregated within one tissue or across tissues in your cohort. Geometric profiling was utilized in David Rimm's Nature Communications article in 2020. Many users can also create rules for segmenting the ROIs. This would let morphology of the tissue determine the boundaries of the area of illuminations, or AOIs, to enable 100% cellularity. For example, users can create tumor and TME segments across a tissue sample as shown on this example. Selected ROIs are illuminated with UV light to release barcodes from DSP probes. And then these DSP probes are deposited into a 96-well plate. The released DSP barcodes are prepared via a standard library preparation, so they can be read out on Illumina sequencers. The data can be mapped spatially back to the originating region of interest. Samples can now be loaded onto your Illumina sequencer. Depending on your experimental design and the instrument capacity, you can read up to 760 ROIs from a single Geomix experiment in a single Illumina run. Here we have an XG 2000, which will allow us to easily read hundreds of protein or RNA analytes. Once the FASTQ files are generated, they'll be converted to digital code counts and then uploaded back to Geomix DSP instrument. The data analysis screen is divided into 3 panes and a taskbar. Here we describe the functions of each pane and the interactions between all 3. The scan pane is shown on the left side and contains individual viewers of the scans. In the middle pane, we have our datasets and probe lists. On the right, we have our visualization pane, which shows the various graphs. In this case, this is our QC graph. The middle datasets pane lists all datasets and probe groups associated with the current study. The visualizations pane is on the right. Visualizations are visual representations of the dataset selected and probe selected. and the adjustments applied to the data set from those probes. Now I'm going to open another study. Here we can see the data has been QC'd and normalized. Once you normalize the data, you can see in your visualizations pane different graphs, including pathway maps, pathway tables, bar graphs, correlation plots, box plots, PCA plots, trend lines, and we can view heat maps. I'm going to end with clicking on a data point on the data visualizations pane, and you'll notice that ROI associated with it will be zoomed in on the left pane of the screen, presenting a seamless integration of the images and the digital counts. Thanks for joining our Customer Experience Lab sessions.